All right, welcome to part 3. We're going to examine damage to the genetic code as a variety of causes and of effects. And this is perhaps a very important aspect of the genetic code that really we have to understand the effects of radiation and various other things. So causes and effects of mutation. Alterations of the sequence of bases in DNA can lead to changes in the structure and function of proteins produced and can have a range of effects. With transcription translation, organisms can convert the information held within the genes into proteins necessary for life, but the process is only as good as the underlying genetic information, and sometimes something occurs that alters the sequence. Such an alteration is called a mutation and can lead to changes in structure and function of the proteins produced. A normal fruit fly, a mutant fruit fly, and you can see a slight alteration there. Mutations can have a range of effects. Sometimes they result in serious, even deadly problems for the organism. Sometimes they have little or no detrimental effect at all. And occasionally, but very rarely, they may even turn out to be beneficial to the organism. So breast cancer in humans, there's 2 human genes called BCRA1 or BRCA1 and BRCA2. More than 200 different changes in the DNA sequence of these genes have been detected, each of which results in an increased risk of developing breast cancer. So mutations have a bad reputation, tend to be disruptive, and are very, very rare. So the types of mutations we have: base pair substitution, base pair insertion, and base pair deletion. And so in terms of the chromosome, the gene deletion, we can have a whole deletion of a gene relocation, movement of the gene to a different chromosome, or gene duplication. So these are the different types of mutations that can occur. And there are certain decisions that we make in life, smoking, getting zapped by radiation of the sun, or getting X-rays or exposure to radiation can affect this mutation rate. So why is it dangerous around the core of a nuclear power plant? Well, mutations are alterations of a single base or changes of large segments of DNA. They are usually rare and occur in genes usually disrupt a normal physiological function. They play an important role in evolution. Faulty genes coding for faulty enzymes can lead to sickness. How can people respond so differently to alcohol? A single difference in a single pair of bases in their DNA. Fast flush response. When we consume alcohol, our bodies start a 2-step process in which alcohol molecules are converted from their intoxicating form to their innocuous molecules. Each of the 2 steps is made possible by a specific enzyme whose instructions are coded by the DNA. Fast flushers like Isabella complete the first step of breaking down alcohol, but cannot complete the breakdown because they carry a defective genetic instruction for making aldehyde dehydrogenase, the enzyme that makes possible the second step of the process. So the poison substance accumulates. And the symptoms of the fast flush reaction are due to the substance toxic effects in the body. So you can see these sicknesses, fast flush, for example, or Tay-Sachs disease is another example. Why do many Asians have unpleasant experiences associated with alcohol consumption? Perhaps approximately half of the people living in Asia carry a non-functional form of the gene for aldehyde dehydrogenase, which is a mutation that confer a greater benefit than harm. In a study of 1,300 alcoholics in Japan, not a single one was a fast flusher, even though half of all the Japanese are fast flushers. The minor change in the genetic code that make alcohol consumption unpleasant experience helps the Japanese and other Asians resist the lure of alcohol. In many other cases, perhaps in the majority of genetic diseases, the link between a particular DNA sequence and physical illness is equally direct. Tay Sachs disease, an individual inherits genes with a mutation that causes the inability to produce a critical lipid-digesting enzyme in their lysosomes. This cellular garbage disposal, as you might think. Because these organelles cannot digest certain lipids, the lipids accumulate undigested, and the lysosomes swell until they eventually choke the cell to death. Within the first years of life, this occurs numerous cells and ultimately leads to death, which is not a good thing. From mutation to illness is just 4 steps. A mutated gene codes for a non-functional protein, usually an enzyme, and the non-functioning enzyme can't catalyze the reaction as normal as it would, bringing it to a halt. Although the details differ from one case to another, the overall picture is the same when it comes to many, if not most, inherited diseases. The pathway from a mutation to illness includes these steps. The molecule within in which the enzyme would have reacted accumulates like a blocked assembly line. Then the accumulation or the accumulating chemical causes sickness because it builds up and the concentrations get higher than it would normally. So this would build up and then cause whatever the issues are. So the fact that most genetic diseases involve illnesses brought about by faulty enzymes suggests some strategies for treatment. These include administering medications containing the normal functioning version of the enzyme, like lactose intolerant individuals consume an enzyme lactase, which gives them for a short period the ability to digest or drink some milk. Alternative lactose intolerant individuals can reduce their consumption of lactose containing foods to keep the chemical from accumulating. So those are some ways that we can live with these types of limitations. Most genetic diseases result from individual mutations that cause a gene to produce a non-functional enzyme which turns or blocks the functioning of a metabolic pathway. So this is the most important part of the function of the metabolic pathway. So if we keep that in our mind, that if we have an enzyme that's catalyzing a reaction to the next product step, if you recall in class we talked about enzymes, think about at that step then the buildup of its substrate would start to increase because the enzyme is not working on it to make the product. So that is pretty much what we had talked about prior. So I guess we're back at the we'll be hitting the next portion of this in part I guess we're in part 4 already. So I'll see you in part 4 and study this section here, and we'll move on.